You are welcome back to Cell and Tissue Organization course. In our previous session, we looked at how nerve impulse is generated. So under session 4 of Unit 5, we will look at the propagation of action potential. That is how nerves are able to transmit messages from one point in the body to another point in the form of an action potential. We also look at the relationship between the strength of a stimulus and the impulse generated and the role of the myelin sheath in propagation of action potential will also be explained. By the end of this session, you should be able to explain the movement of action potential along an axon. Also explain the difference in propagation along myelinated and unmyelinated fibers, and also explain the refractory period and the all or nothing law, and finally describe the role of Schwann cells in conduction of nerve impulses. When an action potential in an excitable cell, such as the neuron, is generated, how is it then propagated along the axon? So we will look at the movement of action potential along the axon, and we make use of the local circuit theory of action potential propagation as shown in the figure in this particular slide. When the membrane of an unmyelinated axon is depolarized, the active stimulated zone becomes more positive inside the membrane and the outside of the membrane becomes more negative. So this particular excited zone or activated zone will lie alongside an area of membrane that is also still in the resting stage. So the distribution of charges in this particular state results in a small current that will flow between these areas, that is the depolarized and the resting area. This local current flow will cause sodium channels to open in the resting area of membrane, and the action potential will therefore move along the axon. The current will then flow progressively or smoothly along the axon. In the course of transmission of action potential along an axon, a second action potential could be generated when a second stimulus is given immediately after an action potential has been transmitted. And this is because the sodium channels will still be in their inactive state and cannot reopen until the membrane has returned to its resting potential. So the interval that is between, or the duration which is impossible for the excitable cell or the neuron to transmit another action potential, is referred to as refractory phase or refractory period. Because of this, action potentials are only propagated in one direction. From our previous discussion, we established the fact that in order to generate an action potential, an excitable cell needs to be exposed to a stimulus of a certain minimum strength known as the threshold. However, any stimulus above the threshold will also elicit an action potential, and the impulse will always be of the same duration and size irrespective of the strength of the stimulus. And this particular phenomenon is referred to as all or nothing law of impulse transmission. We've also studied that many axons are surrounded by a multi-layered white phospholipid covering called the myelin sheath. Now let's have a look at the effect of myelination on axons. The myelin sheath functions to insulate the axon and also increase the speed of nerve impulse conduction. So in the peripheral nerves, these myelinated sheaths are provided by Schwann cells. And in peripheral nervous system, the outer layer of the multiple wrappings is referred to as neurulema, and it is important in regeneration of injured axons. And between the segments of the myelin sheath are the unmyelinated gaps called the nodes of Ranvier. The sodium-potassium channels are much more concentrated at this unmyelinated portion along the axon referred to as the node of Ranvier. And so the action potential is propagated by a series of jams between the nodes of Ranvier, and the leaping from one node to another is referred to as saltatory conduction. And in this way, the myelination increases the speed of conduction. This brings us to the end of this session. So under session 4 of Unit 5, we've learned about the movement of action potential from one section of the axon to the next. And we also covered the all or nothing law. We also looked at the refractory period or phase, and then the effect of myelination.